for the final part of chapter 4. And if you're wondering why it's so long, it's because we are only doing chapter 4 this week. So it is for lectures on 2 days. So that is 3 hours of lecture in a week. This is it. This is the last lecture for the week. Eukaryotes. Okay, so we're going to look at how eukaryotes are different from prokaryotes. Now, we will not be going as in depth on eukaryotes as we did with prokaryotes, and part of the reason is that there's just too many differences between the different kingdoms of eukaryotes. So we will see and learn even more detail about eukaryotes as we study them in our overview of diseases from fungi, helminths, protozoa, etc. Okay, so this won't be as long. All right. So remember, if you see a nucleus or if you see organelles, if we're talking about organelles, that is automatically a eukaryote, right? Prokaryotes do not have a nucleus and they do not have organelles. Prokaryotes do have ribosomes and they do have DNA because those are not membrane-bound organelles, right? Okay. So we talked about a lot of possible structures on the outside of a prokaryote, and one of the things we talked about was flagella. And I want to point out that for the few eukaryotic organisms that have flagella, it is a more advanced flagella. It's much larger. It's made of microtubules, which are hollow tubes made of tubulin, and then inside of the microtubule you'll even find cytoplasm, and then there's a membrane that envelops the whole thing. So the flagellum of a eukaryote is much more advanced and larger, and they even move differently, okay? And they can move much more precisely. And if you are trying to relate this to things you already know, when you studied the cell cycle and you saw everything double and divide, you usually study cell division in a eukaryotic system, and so you probably saw centrioles, which would then grab onto the chromosome and pull it apart. So those are also a type of microtubule, just to give you a little connection there. And then some eukaryotes also have cilia, and you might be familiar with cilia because your lower respiratory system is lined with cells that have cilia, and the function of that is to propel mucus up and out of your lungs so that you swallow it constantly. And this is how you clean your lungs, right? There's all sorts of junk in the air. You're constantly breathing it, and you need to be able to keep your lungs free of debris and free of microbes, because you need gas exchange to happen in your lungs, and it won't work if it's full of other stuff, okay? So cilia in our lower respiratory tract is responsible for keeping our lungs clean and clear of debris. Also prevents a lot of infection because everything gets trapped in the mucus, and then we propel the mucus up and out. Other eukaryotic examples of cilia would just be a unicellular organism that has cilia and uses it to actually move through an aqueous solution. Okay, so we learned that flagella in the eukaryotic organism is much larger, much more complex, and it's made up of microtubules, which are hollow tubes called tubulin. Tubulin is a protein, so it's made of amino acids, right? I related that to centrioles, are also made of microtubules. Centrioles are the paired barrel-shaped organelles located in the cytoplasm of the animal cell, located near the nuclear envelope, and then The centrioles play that key role in extending microtubules throughout the cell to provide the cell's skeletal system, but they also play a key role in forming centromeres for grabbing onto chromosomes and pulling them apart during cell division. Okay, and one more thing to relate to that you probably already know about, actin and myosin, which are located in all of your muscle fibers, that's what composes your muscle fibers, those are a type of microfilament, which is thinner than a tubule, right? It's not hollow, so it's microfilament, which is also a protein made up of amino acids. Okay, so there you go. Now, right, so remember the eukaryotic flagella has the cytoplasm. The entire flagella is covered by a phospholipid bilayer, a membrane, right? And it will move in a wave-like motion instead of corkscrew like we saw with bacteria. And not all eukaryotic cells will have flagella, and not all eukaryotic cells will have cilia either. And in fact, an easy way to remember that is that humans only have one cell with flagella, which are sperm, right? Only men have that. So not even all humans, just about half of humans have one cell type with flagella. So definitely when we learn these things, you need to keep in mind that we're just talking about sometimes this structure appears when we're talking about flagella. When we talk about organelles, then that's usually, for the most part, all cells will have the same organelles, okay? But even that, we'll see some differences. So see this vacuole, this really large organelle here. It could be full of water or it could be full of gas, right? And I talked to you a little bit about if you had an aquatic organism, right, a single-celled organism that's aquatic, it would need to be able to raise and lower its position in the water, and it can do that by letting in more or less air into its vacuole, right? So maybe food is on the surface, but maybe also predators are on the surface, so it needs to be able to go back and forth. Plants, on the other hand, fill their vacuole with water, and that's why when a plant has gone too long without being watered, its leaves get kind of droopy. It's because those vacuoles are now empty. And then, somewhere from an hour to a few hours after you water the plant, you see it perk back up because those vacuoles will fill back up with water. So only cells of, plants and aquatic organisms have these vacuoles. Another thing that only plants have, which I put in green, are chloroplasts, right? So chloroplasts have the green pigment, chlorophyll, that perform the special process of taking sunlight and turning it into sugar. And so that's photosynthesis, right? We don't find that in other eukaryotic organisms like animals or protozoa, okay? Most, if not all, cells will have the rest of these organelles, and we will briefly go through. So make sure that you maybe blank these out and test yourself if you can name all the parts of the cell, and then also make sure you can define the function of all of those. So we'll briefly go over some of those functions, but make sure you read in your book so you have a complete understanding. Okay, so the eukaryotic cell wall, we're going to start with the cell wall and then we'll go through the different organelles. Okay, so the cell wall is not all cells that are eukaryotic will have a cell wall like you and I. None of our cells have cell walls, right? Animal cells do not have cell walls, but fungi do, and their cell walls have a characteristic combination of cellulose and this chitin protein. It's actually a carbohydrate, sorry. So chitin, carbohydrate, cellulose, Glucan, another type of carbohydrate, and mannan, another type. The predominant characteristic of fungi is chitin, okay? Plants also have cell walls, and of course their characteristic cell wall component is cellulose, right? And algae we don't really talk about in this class, but they'll have, they can have glucan, mannan, or cellulose. But it's mostly just fungi that have chitin, and plants predominantly have cellulose. All right, so unique carbohydrate types. The glycocalyx that we learned about for bacteria also exists in animal cells. We excrete, but then we call it an extracellular matrix. And this is used to help make tissues, so bonded to proteins and lipids in the membrane. And this can just help us to be able to communicate with other cells of a tissue, create special environments. You may have learned about the extracellular matrix if you have already taken physiology. You can read a little bit more about that in your book. Okay, so no peptidoglycan in any eukaryotic cell, right? Only bacteria cell walls. So it looks very similar to, the membrane will look very similar to the membrane of bacteria, but instead of having those simple haploids, you'll see more complex sterols, and in animals you'll specifically see cholesterol, but it's doing the same job and giving extra structure and support to the membrane. You'll see more peripheral proteins, more integral proteins, transmembrane proteins, right? Because you just have a lot more activity, a lot more complex interactions going on. So bacterial cells that are capable of respiration, so instead of only fermentation, they're able to have an electron transport chain. Bacterial cells don't have organelles, so they don't have a mitochondria to do that. They have to use their membrane. So you'll actually see a whole bunch of the proteins for the electron transport chain in their membrane, where in eukaryotic membranes you wouldn't see that because we have a whole bunch of mitochondria that have all the membrane space to do that, and it makes it more efficient for us. Okay, so that concept will come up again when we look at metabolism. So a little bit more about the membrane. It can be used to bring large particles in or release large amounts of things out. So we see endocytosis is the process of bringing some substance inside of the cell. And we have three main mechanisms to do that. Phagocytosis happens only through phagocytic cells, such as our white blood cell phagocytes. Some of our white blood cells in our immune system are phagocytes, and they extend their membrane in as if they're extending these hands or feet to grab and bring in the substance. So phagocytes in our immune system do this to take up possible invaders, such as bacteria, and destroy it, while keeping it inside of this vesicle to destroy it. Other eukaryotic organisms like amoeba extend these pseudopod feet-like structures to engulf a bacteria or other particle to use it as food, right? So same overall mechanism of engulfing and keeping in a vacuole, but one is to destroy and get rid of, and one is to eat as food, right? We also have penocytosis, bringing in small substances by in what we call invagination or inward folding of the membrane, which also leads to the formation of a vesicle, which will contain some of the extracellular fluid and the substance. So penocytosis, the membrane folds inward, right? Bringing in fluid and dissolved substances, which then could be used for food or whatever the cell needs to can check out what it is and use it. Then we have a little bit more fancy process, which is similar to pinocytosis but more specific. It's a receptor-mediated endocytosis where some substance in the outside world is able to specifically bind to receptors on the membrane, and that causes it to fold in upon itself and make a vesicle. And this vesicle is going to be coated because of those receptors in the membrane. And so you'll see it's a specialized vesicle. The cell will have a good idea of what's here because it had to have been able to specifically bind to the receptor. And that's really a good introduction. We see a lot of receptor-mediated endocytosis when it comes to mammalian viruses, for example, that take advantage of our receptors on certain cells. So you'll see that again. All right. So this is the picture I was telling you about where we can compare eukaryotes and prokaryotes. The size is huge. The difference in size is huge. So this is one cheek cell, so it's a eukaryotic cell, and this is its nucleus. Whereas all these little dots that are kind of purple dots that you're seeing, those are bacteria, prokaryotes. So see how tiny the entire cell is, just a dot smaller than the nucleus of the eukaryotic cell. So huge, 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 huge difference in size, right? So the eukaryotic cell is about, I don't know, 30 to 50 micrometers, whereas the prokaryote is like 1 to 2. So huge difference. All right, now not every eukaryote will be exactly this huge, and not every prokaryote will be exactly that size, but that's about what you'll see, right? Okay. You already know the cytoplasm is the substance inside of the plasma membrane and outside of the nucleus, and the cytosol is the fluid portion of the cytoplasm. Okay, that hasn't changed. Those are the same terms. Cytoskeleton, now we have microfilaments, intermediate filaments, microtubules, just more advanced structure. Structure and support to the cell gives the cell its shape, also helps carry out a lot of functions, like the centrosome, which we were starting to talk about already, microtubule organizing and construction center, key role in cell division, so lots of activity going on. We also have ribosomes, but they are a little larger. They are made up of an 80 and a 70. So protein synthesis, ADS, membrane bound, attached to the ER. Sometimes they're free in the cytoplasm, whereas we will have the 70S, which is the same as the bacterial ribosome, in our mitochondria. But the vast majority of our ribosomes will be the ADS, which will either be membrane bound on the ER, which we then call rough endoplasmic reticulum, or they can be free floating in the cytoplasm as well. Okay, so the majority of our ribosomes, again, are ADS, which are larger. And this is why we can have those antibiotics that we looked at that target the bacterial ribosomes. Because our ribosomes are different, we can attack bacterial ribosomes without hurting our own. Okay, so whenever you think about drugs, you have to make sure that the target is specific to the pathogen, and we will not be targeted and get harmed. Okay, so organelles, that you remember, organelles are only found in eukaryotes. The nucleus is actually a double membrane organelle, which we'll see in a moment. It contains our chromosomes for DNA, and it's also the place where mRNA is made to make proteins. Okay, the ER stands for endoplasmic reticulum. It's a network where transport happens, and a lot of protein synthesis happens there, which is turning that mRNA transcript, which is a nucleic acid, and it turns it into amino acid sequence to become a protein. Okay, the Golgi complex is also a membrane kind of network formation that is responsible for a lot of secretion. So if you need to release something from the cell, it needs to have a membrane. The Golgi apparatus is usually where it will get the membrane from. And so go ahead and take a look at that in your textbook. Lysosome, we talked about that enzyme that's able to digest the peptidoglycan. And we have, so lysozyme is the enzyme, is the digestive enzyme. So the lysosome is a vesicle that contains a bunch of the digestive enzymes. And then the vacuole, I talked to you quite a bit. It can be used for food for some organisms and used to for water or gas for other organisms. And again, you do not find a vacuole in every single eukaryotic cell. The mitochondria I started to talk to you about, it's the site of cellular respiration, the last two parts of cellular respiration, where you'll have the Krebs cycle, and then you'll have the electron transport chain. And the electron transport chain is a series of proteins embedded in the inner membrane. So the mitochondria also is a double membrane organelle, and that's where all of our ATP is produced for our cell. Peroxisome is another defensive type vessel. It will be able to carry out the oxidation of long fatty acids into shorter ones. And then short fatty acids go to the mitochondria to be used to make ATP. But at the same time, peroxisomes can be used as a defense to oxidate bacteria and kill it. Okay. Also, we have the ability to, and I don't know why I put destroy hydrogen peroxide. It's kind of a weird way to put it, but break it down into water and oxygen. So the eukaryotic nucleus, as I told you earlier, is a double membrane. So you see this nuclear envelope, double membrane, and then there are specialized pores throughout so that we can get materials in and out as needed, but we are being very protective. We protect our DNA. The DNA in this image is has been replicated and condensed, and it's now ready for cell division. And otherwise, you wouldn't be able to see it dense like this. It'd be spread out and being used to make copies of genes to make proteins. When it's condensed like this, it cannot be accessed. So that's how we know that this is about to go through cell division. Okay, the region of this condensed DNA is called the nucleolus. Okay, how is it condensed into this super tight form? Through histones, special proteins used to wrap DNA into a tight condensed form. So these really neat processes that we talked about, you can see nice visuals with these videos. These are all really great. They're not too long, and I highly suggest you watch at least one, the first one or the second one, because they give you cell structure visuals with function review. Okay. So make sure that you understood the significant differences between eukaryotes and prokaryotes, specifically flagella, cilia, cell walls, plasma membranes. I don't know why cytoplasm is written on there. The cytoplasm's mostly the same. You'll just find a lot more stuff in the cytoplasm of a eukaryote. And then remember we discussed the antibiotic erythromycin that binds to the 50S portion of the bacterial ribosome in the prokaryote, right? But on the eukaryote, we have a different ribosome, and so it's not going to harm us. Okay. So I've started a compare and contrast for you. I would like you to complete it to help you study. So prokaryotes have one circular chromosome. Eukaryotes, on the other hand, have individual chromosomes that are not circular, and it's more than one chromosome, and it depends on the organism. You don't find histones in prokaryotes. They're not that advanced. They don't have any organelles, no membrane-bound organelles. Bacteria will always have peptidoglycan in the cell wall, whereas archaea will have pseudomerin if they have a cell wall at all. Eukaryotes have paired chromosomes in the nuclear, located inside of the nuclear membrane. That's another way of saying inside the nucleus. Okay, and of course we do have histones that condense our chromosomes. We have lots of membrane-bound organelles. Make sure you know them. And we have, if there's a cell wall, there are different types of polysaccharides for different types of eukaryotes that we looked at, and make sure that you can name those. Okay, so that's it. You are done with chapter 4. Make sure you read it. Do the practice questions at the end of the chapter. You can also use these little check your understanding questions throughout the slides. Come to office hours if you have any questions. And that's it. Have a good day.